Now, here we go. All right. There's one more complication in studying variance, and that's something that we left out. Some variance results not from the environment, not from genes, but from interactions between genes and the environment. There are lots of cases where genes turn on, are activated, or are silenced, or their expression is modified because of some environmental cue. Now, I had an example of this that a few minutes ago was running through the house and getting into an all out fight with one of our chairs, but she's left now. One of our zoo of cats, named Tsubaki, has the Siamese phenotype. She's not a pure Siamese, but she's got the Siamese pattern, which, as you remember, I trust, is dark pigment on the cooler parts of the body, right? Siamese cats are, most of the body is light colored, but they'll have dark faces, dark ears, dark feet, and a dark tip of the tail, with the rest of the body being fairly light. That is because in Siamese cats, the pigment gene is only switched on when the temperature is just a little bit cooler than core body temperature. The upshot of that is that if you take a Siamese cat and you shave off a patch of its hair and you tape an ice pack to the shaved part, what you'll end up with is an extremely pissed off cat that craps in your shoes. But you'll also end up with a cat where the hair will grow back darker in color. I've never tried this, but my wife, when she was little, the family had a Siamese cat that spent a lot of time outdoors. And she's told me that that cat got measurably darker in winter than in summer. When that outdoor cat was exposed to cold temperatures, her fur came in darker, and she would lighten up in summer when the temperatures she was exposed to were warmer. That's an example of gene cross environment interaction. Just how dark that cat was neither entirely due to genes nor entirely due to environment. It was due to the ways in which genes and environment interact. And the description of environment gene interactions is called a reaction norm. Genotypes often don't specify exactly what the phenotype is supposed to be. Um, my wife's Siamese cat did not have genes specifying exactly how brown her fur was supposed to look. She had a genotype that determined a range of phenotypes that the cat would have under different conditions. The genes determined the range, and then the environment that the cat was in, sort of picked within that range what the cat's actual color was going to be, whether it was going to be more dark or a more light shade. So the fact that phenotypes vary with environment has a name. We call it phenotypic plasticity. Plastic, again, plastic is the material, but plastic as an adjective just means moldable, shapeable, because plastic, the material, is called that because you can mold it into any shape that you want. So phenotypic plasticity is like the ability to mold the phenotype. And the mathematical relationship between phenotype and environment for any given genotype is called the reaction norm. Let me see if I can clarify this with an example or two. These are rotifers. You may or may not, those of you that went through invert zoo, we tried to look at rotifers, although I'm not sure how well that lab worked when we were doing it remotely. But rotifers are animals. They consist of several hundred cells. They are microscopic or very close to it. Some of them settle, many of them swim, and this is one called Brachionis caliciflorus. Now, on the left, that is a swimming Brachionis caliciflorus. The head is at the top. There's kind of a tail at the bottom, and surrounding the whole thing, there is a firm covering that's got a definite shape. And you can see the very symmetrical shape of the covering, the cuticle, right there. That's Brachionis that's being grown in an environment where there's no predators. If we were to add some predators, like the big rotifer Asplankina that eats Brachionis, we would start seeing Brachionis sprouting spines. You can see them over there on the right. That's what Brachionis looks like when predators are present. In fact, Brachionis will grow those spines when there's no predators, but you expose it to water that the predators have been living in. Brachionis responds to some kind of chemical that the predators send out and sprouts those spines to make it more difficult for the predators to capture and eat them. That's phenotypic plasticity. The phenotype of Brachionis changes depending on what's in the environment, whether there are predators or not. Some species of Daphnia do the same thing. Over there on the left, that is a scanning electron micrograph of an ordinary Daphnia. This is the so-called water flea. Very common freshwater, tiny crustaceans, maybe a millimeter long, maybe a little more, but not much more. On the right, that's what an ordinary Daphnia looks like. On the left, that's a Daphnia grown in the presence of predators. Specifically, I think it's midge larvae, if memory serves. And you can see the Daphnia on the left has grown a big tail spine and a head spine. The Daphnia on the left looks like it's wearing a helmet. That's another case of phenotypic plasticity. Specifically, this is often called an inducible defense. The spines cost metabolic energy to grow, right? Daphnia, it's not efficient for Daphnia to grow these if there are no predators present. So Daphnia has adapted to only grow those spines in the presence of predators, specifically responding to chemicals that the predators put out, chemicals called pheromones. So that's a case of phenotypic plasticity. The genes don't specify one phenotype. The genes specify possibilities, and then signals from the environment determine which possibility actually shows up. This is a neat example. This is a European wildflower called Ranunculus aquatilis, water crowfoot. The ranunculus that we get around here is the buttercup. We have lots of buttercups in, grassy areas and unmowed lawns and things like that all over the place. Well, this is an aquatic relative of theirs. And it tends to grow right on the edges of streams, which means that at any given time, you'll have part of the plant growing under the surface of the water and part of the plant growing above. The part of the plant that's growing above has these broad leaves. The part of the plant that's growing below the surface of the water, the leaves are little bundles of filaments. You can see down here at the lower right, that's what the leaves look like underwater. Up at the top and left, those broad leaves are what the leaves look like when they're in air. This is on the same plant, so there's no genetic difference, right? All of the cells in this plant are genetically identical, except for a few mutants. But the genes in this plant must give it the possibility to select one of two phenotypes either the broadleaf or the filamentous leaf, depending on what environment any given part of the plant is growing in. Now, Lamarck actually mentioned water crowfoot and several other examples and used them as support for his theory of evolution by the inheritance of acquired traits. And he's quite right in that phenotype can be altered, sometimes profoundly, by the environment that you're in. Oh, yeah. This is an example you might actually still see. I took this picture in Kroger. I think that's the bougie Kroger over on Salem Road. A couple of springs ago, they were selling hydrangeas, and um, they're a popular garden plant. My mom has a bunch; she loves them. Maybe you do, or your mom does too. And the common hydrangea garden species are phenotypically plastic. This is not an inducible defense, but it is phenotypic plasticity. If you plant one of these in acidic soil, acid soil has free aluminum ions. Aluminum doesn't bond to soil particles. The aluminum is soluble. The plants can take them up, and the plants use aluminum to produce blue pigment. So the plants on the left are growing in slightly acid soil. They look blue. The plants in the middle, they're growing in more alkaline soil. Alkaline soil does not have free aluminum. The aluminum tends to bond to soil particles if it's there. Those plants can't take aluminum up, and they don't make blue pigment, and they end up pink. So the difference between blue and pink hydrangeas is phenotypic plasticity. In fact, you can plant one and turn it into the other by fiddling with soil chemistry, by adding soil additives that will turn it more acid or turn the soil more acidic or more alkaline. Incidentally, the plants that are white over there, that's not phenotypic plasticity because those plants have mutant alleles that don't make any pigment at all. So white hydrangeas are not an example of phenotypic plasticity. Their phenotype is pretty much fixed because they'll never make any pigment no matter what kind of soil you put them in. So for a lot of species, you can have this mixture of traits being phenotypic plasticity in some situations and not plastic in others. So there. They might still have some of those. I usually see them in spring. It might be a little late, but I don't know. You could go see this if you wanted to. This is what I mean by a reaction norm. A reaction norm is a mathematical description of phenotypic plasticity. What's graphed up here is down on the x-axis, you've got temperature. On the y-axis, on a log scale, you have the number of facets in fruit fly's eye. So somebody raised a bunch of fruit flies and somebody counted the number of eye facets, probably a grad student. And up here at the top, you've got wild type, just, regular flies that you might catch in your kitchen if you left a bunch of bananas out for too long. And this trait is phenotypically plastic. If you raise your flies at 15 Celsius, you have about 1,000 facets per eye. That number goes down with increasing temperature until when you raise your flies at 30 Celsius, that's what, 85 Fahrenheit or so, you have maybe 700, 800 facets. So the number of facets is actually temperature dependent. These are fruit flies with two different mutant phenotypes called infrabar and ultrabar. They both produce a phenotype with unusually small eyes, called bar eyes. Much fewer facets. The eyes are not the big globe-shaped things that you would expect in a typical fly. They're more like little strips. And at about 17 Celsius, where these lines cross, both infrabar flies and ultrabar flies only produce maybe 180 facets per eye. The difference between infrabar and ultrabar is not that they're always phenotypically different. Under some circumstances, they produce the same number of facets, but flies with infrabar, the number of facets goes up a little bit with increasing temperature, and flies with ultrabar, the number of facets goes down all the way to about 70 if you raise these at the highest temperature of 30 degrees. So these are two different alleles. They're not so much different because they produce different phenotypes, because under some circumstances they produce identical phenotypes. What's different about them is that they create different reaction norms, right? They respond to the environment in different ways. Okay, going back in time a little bit to 1948, this was a famous experiment done on a plant called wild yarrow. Um, I'm not sure what species this is, but Achillea millefolium is actually cultivated. You can put it in your salads, and it may have some medicinal properties as well. But here's some wild yarrow. Clausen et al. Back in 48 um, took 7 wild yarrow plants and grew cuttings from them in the same garden at a site in California called Mather. And here's what they got. They're all growing in the same environment, right? They're all growing in, this is again what we call a common garden experiment. They're all in the same garden. They've all got the same soil, um, the same amount of light, the same amount of water, the same temperature fluctuations. They're all growing as identical in environments as identical as possible. So that means that we have no environmental variance. So the differences between these plants have to be genetic, right? And you can see plant A is growing very tall, plant E is second, plant G is not really going anywhere. All of these have to be caused by differences in genes, right? They grew cuttings from the same 7 wild plants in a different garden under different conditions. Mather is up in the Sierra Nevada mountains, but they grew cuttings from the same wild plants down near San Francisco Bay, down at Stanford, California. Much lower elevation, much milder climate. I mean, it still gets cold and foggy in the mornings, but not nearly the kind of rough climates that you get up in the Sierra Nevada mountains at maybe, I don't know, 7,000 feet or however much it was. A lot more moisture from dew, damper climate, and so on. And under those circumstances, they got this. Now, again, all 7 of these plants are growing in the same environment, so the differences between them must be genetic, right? There's no variation due to environment in this case. And here we put these two together and notice something. In Stanford, A did the best, B came in second. Up at Mather, B did not do very well, and E came in second place. A managed to win both, but E did a lot better at high altitude. It didn't do quite so well comparatively at lower altitude. C came in third up here. You can see B and D didn't do very well, but B and D both got very tall down at Stanford. Um, what's different about these plants is their norms of reaction. Okay, remember that each of these plants, like the A here and A there, these are genetically identical. These two, this one and that one, they're genetically identical because they're cuttings. They were grown from cuttings off the same plant. That plant and that plant are genetically identical because they're grown from cuttings and so on. So what's different about these 7 different yarrow plants is they have different norms of reaction. It's not so much that they're different in phenotype, they're different in how they can make the phenotype respond to being found in different environments. And remember, the mathematical description of how phenotype responds to different variables in the environment is a reaction norm. So what's different about these 7 yarrow plants is they have different norms of reaction. Okay, actually, let me check the time. Do I want to get into that? All right, I'll tell you what, I think I may actually be missing a couple of slides, so I'm going to go ahead and take a pause. And I'm going to go and stop recording, and I'm going to see if any of my slides have gone missing. So I'll stop the recording now.